Welcome back to my channel. In this video we're going to go over the structure and function of cilia and flagella. Cilia and flagella are both extensions of cells that are involved with movement. There are three types of flagella, those found in bacteria, those found in archaea, and those found in eukaryotes. In eukaryotes, both flagella and cilia are structurally similar. They are both extensions of the cell membrane. Within the flagella or cilia is the cytoskeleton, which is called the axoneme or axial filament. This internal cytoskeleton is made up of microtubules. In eukaryotes, the microtubule cytoskeleton consists of 9 fused pairs of microtubules that form a circle around the periphery of the core of the flagella or cilia. In the middle of this are 2 central single microtubules. This creates a 9+2 arrangement of the axoneme that is the typical arrangement in eukaryotes. The eukaryotic flagella or cilia also has a basal body at the base of the extension. The basal body is a short cylinder with a slightly different cytoskeleton. In the basal body, there are 9 microtubule triplets forming a ring around the periphery. This forms a 9+0 arrangement. The basal body is separated from the rest of the extension by a basal plate. How are eukaryotic cilia and flagella different from one another? The first difference between the two are that they have different lengths. Cilia are short, whilst flagella are longer and thread-like. The second difference is how they move. Cilia move in a wave-like motion, beating back and forth. Flagella, on the other hand, move in a more rotational movement, similar to how a propeller moves. The third difference is in their function. Cilia function in locomotion when found on unicellular eukaryotes. In multicellular eukaryotes, cilia function together to move substances across the surface of the cell. Flagella primarily function in locomotion, moving an entire cell around, such as moving around a single-celled eukaryote or a single cell such as a sperm cell. The second type of flagella are those found in members of the bacteria, which have whip-like flagella. The bacterial flagellum has 3 regions: the basal body, the hook, and the filament. The filament portion is helical in shape and is the longest region of the flagellum. This filament is a hollow tube made up of flagellin proteins. The basal body is the portion that is embedded into the cell wall. The hook is a short region of the flagellum that has a sharp bend and joins the filament to the basal body. The basal body is the most complex region of a bacterial flagellum. It consists of a series of rings and a central rod that extends into the hook. These rings and rod act like a rotor to move the flagella. Gram-negative bacteria have 4 rings in their basal bodies that are connected to a central rod. The outer L ring is associated with the lipopolysaccharide outer membrane. The P ring is associated with the peptidoglycan cell wall. The inner MS rings are associated with the inner membrane. The last ring is the C ring, which sits in the cytoplasmic space attached to the MS ring. The basal body is responsible for the rotor movement of the flagellum filament. It is able to rotate the filament both clockwise and anti-clockwise to create movement in the bacterium. Gram-positive bacteria have just 2 rings in their basal bodies. The outer ring is attached to the peptidoglycan layer. The inner ring is attached to the plasma membrane. Growth of the bacterial flagellum occurs at the tip. The function of the flagella in bacteria is primarily in locomotion, causing movement in the bacteria. There are several different types of flagella arrangements in bacteria. When there is a single flagellum extending from one end of the bacterium, it is called monotrichous. When there are 2 flagella, each located at opposite ends of the bacterium, it is called amphitrichous. When there is a cluster of flagella located at one end of the bacterium, it is called lophotrichous. When the flagella are located randomly over the entire surface of the bacterium, it is called peritrichous. The third type of flagella are those found in members of the archaea. These structures are known as archaeal. The flagella of archaea are similar in appearance to bacterial flagella. They both have long filaments extending from the cell and a basal body. As with bacterial flagella, they rotate to move. One difference is that archaea are thinner and not hollow, whereas bacterial flagella are thicker and hollow. Growth of the archaealum happens at the base of it, whereas it happens at the tip in bacterial flagella. Another difference is that in archaea, rotation is powered by ATP, whereas in bacteria it is powered by proton motive force of hydrogen ions. The last difference we're going to mention is that archaealum are more similar to type 4 pili than they are to bacterial flagella. The function of the archaealum, much like that of the flagella in eukaryotes and bacteria, is a movement of the archaea. And that concludes this video on the structure and function of cilia and flagella. Thank you for watching. If you want to support this channel, please subscribe and check out more of my videos.